Ladies and gentlemen, welcome to the Second International Scientific Conference, InterSci 2025, Global Innovations in Modern Science, hosted by the Juraj Dobrila University of Pula. We are gathered here today to celebrate global innovations in modern science. Congratulations to all participants for their outstanding contributions and dedication to advancing knowledge in their respective fields. I wish you all a successful, inspiring conference, filled with productive discussions and groundbreaking discoveries. We invite Mrs. Petra Buric to speak. The topic of his speech is geospatial pattern of topsoil pollution and multi-endpoint toxicity in the petrochemical area of Augusta Priolo, Eastern Sicily, Italy.
Good morning, esteemed colleagues, ladies and gentlemen. My name is Petra Buric, and today I am presenting research focused on a critically important topic: the influence of the petrochemical industry on public and environmental health. The operation of the petrochemical industry continues to draw scrutiny due to its potential contribution to pollutant releases and subsequent adverse effects on human and environmental health. This is particularly concerning because industrial facilities are often located in close proximity to population centers. The global reliance on fossil fuels complicates the transition to cleaner energy, making the rigorous monitoring of health in communities near industrial sites essential. Epidemiological research provides strong evidence linking residents near petrochemical complexes to increased health risks. For example, studies in Asia have revealed a significantly greater incidence of cancers and an overall elevated risk of leukemia for residents living within a few kilometers of major complexes. Furthermore, negative impacts are also documented in occupational settings, with exposures linked to increased concentrations of various metals in workers' urine and elevated risks of specific conditions like miscarriage. In the Italian context, workers in the petrochemical sector have been reported to experience significantly higher incidences of malignant tumors and respiratory system diseases compared to other industries. In Sicily, public health surveillance programs have consistently documented adverse health outcomes. Analysis of congenital anomalies in the industrial area of Gila showed a highly concerning 236% increased incidence of genital defects in newborns during the 2006-2010 period. Similarly, the Augusta Priolo Melilli conurbation, a site hosting 3 major oil refineries and related facilities, representing one of Europe's largest industrial footprints, also recorded high rates, including a 154% increased incidence of genital defects and a 165% increase in digestive system anomalies in newborns relative to national baseline values during the same period. These elevated rates of negative health outcomes are hypothesized to be driven by industrial emissions. Recognizing the clear evidence of contamination and health concerns in the region, an ad hoc investigation was crucial to evaluate the environmental effects of topsoil contamination near the Augusta Priolo facilities. This study aimed to identify the geospatial patterns of pollutants and their complex, mixture-related toxicity in topsoil samples collected near the heavily industrialized Augusta Priolo area, which supports a population of approximately 50,000 inhabitants. The chemical characterization was comprehensive. Elemental analysis was conducted via inductively coupled plasma mass spectrometry, ICP-MS, for 23 metals and 16 rare earth elements, REEs, while organic analyses focused on 16 polycyclic aromatic hydrocarbons, PAHs, and total aliphatic hydrocarbons, C10, C40. Analysis revealed that organic pollutants varied spatially. PAHs displayed the highest concentrations at specific sites, with total C10, C40 hydrocarbons peaking at other locations. To provide a robust toxicity assessment, a multi-endpoint approach using 4 distinct bioassays was implemented, which is necessary given that the results can be model organism dependent. Topsoil samples were tested for toxicity using evaluation of developmental defects and cytogenetic anomalies in the early life stages of the sea urchin Sphaerochinus granularis, the assessment of growth inhibition in the diatom Phaeodactylum tricornutum, the determination of mortality in the nematode Sanorhabditis elegans, and the induction of mitotic abnormalities in the onion Allium cepa. The integration of chemical and biological data established a clear link between contaminant levels and biological effects. Samples collected closest to the defined petrochemical facilities consistently showed the highest levels of select inorganic and organic pollutants, which directly correlated with adverse biological effects across multiple toxicity endpoints. Significantly, the study identified increased levels of total REEs in sites nearest to the industrial complexes. This finding is noteworthy because REEs can serve as valuable signatures, aiding in the identification of petrochemical sources contributing to environmental pollution. In conclusion, the consistent data on soil toxicity, metal, and REE contamination observed across the Augusta Propriolo sampling sites provides a crucial geospatial assessment of risk. This information establishes a reliable baseline for epidemiological studies concerning the high incidences of congenital birth defects reported in the area, facilitating the targeted identification of specific at-risk localities. Thank you for your attention. I am now ready to take your questions.
Invite Mrs. Snizana Babic to speak. The topic of his speech is usability and user experience-related perceptions of university students regarding the use of Bing Chat search engine and AI chatbot. Preliminary evaluation of assessment scales.
Good morning, esteemed colleagues. My name is Snizana Babic, and I am pleased to share the preliminary findings of our investigation into university students' perception and adoption of large language models, or LLMs, in higher education. Since the introduction of powerful AI chatbots like ChatGPT and the more advanced GPT-4, the academic community has been engaged in a continuous and necessary debate regarding their potential impact on teaching and learning. Tools such as Bing Chat, now known as Microsoft Copilot, which integrates a GPT-based chatbot with a search engine, represent a crucial resource in this new landscape, presenting both significant challenges and opportunities for academic instruction. Our preliminary investigation focused on understanding the potential benefits of this tool by assessing specific variables broadly related to the usability and user experience among university students. The study utilized a survey administered to 2 convenience samples of students from different Croatian higher education institutions. We employed assessment scales for several key variables relevant to technology perception, including perceived usefulness, general usability, learnability, system reliability, and, crucially, subjective factors such as cognitive involvement, design appeal, and trust. The internal consistency of these scales was confirmed, lending reliability to our findings. Our data analysis revealed a significant finding. The variables with the highest correlation to the ultimate intention to use Bing Chat or a similar service were cognitive involvement, design appeal, and trust. This suggests that affective and cognitive factors, along with the user's confidence in the AI tool, are more potent predictors of technology adoption among students than purely functional or technical metrics. Overall, students' responses provided a positive evaluation of the various usability and user experience characteristics of the chatbot. These preliminary findings require rigorous discussion within the framework of established technology adoption models. Traditional models like the technology acceptance model primarily identify perceived usefulness and perceived ease of use as the central predictors of a user's intention to use a new technology. This study validates that these classic factors remain pertinent, but critically reveals that for contemporary, conversational AI chatbots like Bing Chat, additional and more subjective variables are becoming paramount in predicting the intention to use. The correlation shift suggests that for AI technologies offering a more interactive experience, the adoption model must be expanded. The significance of trust underscores that because the AI generates information rather than merely processing static data, a student's confidence in its reliability, accuracy, and ethical operation is a prerequisite for sustained reliance. High trust levels directly translate into a greater willingness to utilize the tool for academic assignments. Cognitive involvement highlights that Bing Chat is not perceived as a passive utility but as a learning partner. Students who find the interaction intellectually stimulating and reflective of active learning are more likely to integrate it into their study habits. Lastly, design appeal moves beyond mere aesthetics. In the competitive digital landscape of educational tools, a clean, intuitive interface and positive overall aesthetic experience are now essential for maintaining user engagement and ensuring long-term adoption. These results collectively emphasize that successful AI integration is contingent not only on the technology's technical capabilities but, more importantly, on subjective user perceptions and the human factor. These study conclusions have direct and significant implications for pedagogical design and curriculum development in higher education institutions. Since AI chatbots are recognized as an inevitable force to be integrated into educational practices, educators must transition away from restrictive approaches towards strategies that promote competent and critical utilization. For ensuring successful and sustainable implementation, tasks must be redesigned to prioritize complex interaction over simple fact retrieval. Given that trust is a crucial factor, students must be explicitly trained to critically evaluate AI outputs, particularly regarding common LLM issues like hallucinations. Learning activities should demand information verification against established academic sources, thereby fostering information literacy in the age of AI. To enhance cognitive involvement, assignments should shift from rote memorization to demanding complex, multi-step problem solving where the AI serves as a co-pilot or brainstorming tool, compelling students to critically reflect on the AI-generated responses and apply deep critical thinking. Furthermore, maintaining academic integrity is vital, requiring clear establishment of ethical and procedural guidelines detailing when, how, and for which specific components of coursework AI tools are permissible. This directly contributes to establishing a consistent institutional framework of trust. In summary, the findings indicate that the greatest risk to AI adoption lies not in the technology itself, but in the failure to understand the user's psychological and cognitive interaction with it. Sustainable integration of AI chatbots necessitates a transition toward a user-centric pedagogy where the student's subjective perception, trust, and quality of cognitive experience are the primary design priorities. Understanding these subjective user perceptions, especially factors like trust and cognitive engagement, is critical for ensuring successful and sustainable implementation of AI in higher education. Thank you.
We invite Mrs. Katarzyna Wiktorska to speak. The topic of his speech is guidelines for the use and interpretation of assays for monitoring autophagy.
Good morning, everyone. Since the initial guidelines for standardizing autophagy research were published, our field has experienced rapid growth. Given the expansion of knowledge and the development of new technologies, regular updates to these guidelines are essential for investigators monitoring autophagy across different organisms. A persistent challenge remains the confusion surrounding acceptable and reliable methods for evaluating this complex process, particularly in multicellular eukaryotes. This presentation provides a critical discussion of current methods. Our goal is to assist investigators in selecting and interpreting assays and to help reviewers provide fair critiques of relevant reports. It is important to emphasize that no single assay is universally perfect. The suitability of any technique depends entirely on the specific scientific question and the biological system under study. The complexity of autophagy necessitates the use of multiple, complementary techniques to properly monitor the autophagic process in any given experimental setting. Furthermore, we must be careful with genetic approaches. Because core components of the autophagy machinery have roles in both canonical and non-canonical pathways, genetic approaches to block the pathway should ideally involve targeting two or more autophagy-related genes that function at different steps. Similarly, many proteins involved in autophagy also regulate other cellular pathways, such as apoptosis, meaning not all of them can be used as specific markers for a true autophagic response. Our ultimate goal, collectively, is to foster intellectual and technical innovation within this field. The role of transmission electron microscopy autophagy was first detected using transmission electron microscopy, or TM, in the 1950s. The process is defined by the sequestration of cytoplasmic portions by a double membrane structure called the phagophore, which then matures into the double membrane autophagosome. Subsequent fusion with the lysosome, or the vacuole in fungi and plants, exposes the sequestered cargo to enzymatic breakdown. The importance of TM lies in its capacity to reveal the morphology of autophagic structures at nanometer resolution, allowing for their exact identification within their natural cellular environment. TEM can monitor both selective and non-selective autophagy. However, the sequential morphological changes are continuous and dynamic, which makes discrete classification problematic. For general purposes, the term autophagic vacuole can be used when it is difficult or unnecessary to differentiate between the autophagosomal, amphisomal, and autolysosomal stages. In complex eukaryotic cells, the use of this general term is strongly recommended. Autophagosomes, also called initial autophagic vacuoles, typically show a distinct double membrane visible by Tm. Late or degradative autophagic vacuoles, the autolysosomes, typically possess only a single limiting membrane and often contain electron-dense, partially degraded cytoplasmic material. Because these degradative compartments are often difficult to distinguish from late endosomes or lysosomes even for experts, a simple solution is to group all dark staining structures as degradative compartments. An increase in these structures per cell area indicates enhanced degradation. Unequivocal identification of lysosomes requires immunoelectron microscopy to detect a cathepsin or other lysosomal hydrolase, or structural proteins like LAMP1. Rigorous confirmation requires demonstrating the colocalization of an autophagic substrate and a lysosomal marker, or confirmation of acidification. We must also remember that other processes, such as endosomal, phagosomal, and chaperone-mediated mechanisms, also transport cargo to lysosomes. This means a lytic compartment may contain cargoes from multiple sources. However, the term autolysosome may still be used if the content is overwhelmingly autophagic. In summary, TEM is an extremely informative and powerful method, providing unparalleled subcellular resolution. Its successful application relies on the accurate identification of autophagic structures, which is necessary for reliable quantitative results via morphometry. Ultimately, electron microscopy is best used in combination with other methods to ensure the complex and holistic approach now essential for progress. Western blotting and the ATG8/LC3 conjugation system. The ATG8 and ATG8 family proteins are the most commonly monitored autophagy-related proteins utilized in multiple assays. ATG8 is a ubiquitin-like protein conjugated to phosphatidylethanolamine, or PE. Mammalian homologs belong to two major subfamilies, LC3 and GABARAP. After cleavage, these proteins exist in two forms, the unprocessed form, LC3I, and the PE conjugated form, LC3-2. The PE conjugated form, LC3-2, is the only protein marker reliably associated with completed autophagosomes, although it is also found on phagophores. The conjugation is detected using Western blotting, where the positions of both LC3I and the slightly faster migrating LC3-2 must be clearly indicated. It is critical to specify the isoform being analyzed due to functional and tissue-specific differences. For instance, PINK1 PRKN dependent mitophagy heavily relies on the GABARAP subfamily with minimal need for LC3. A key challenge in mammalian cells is that the total levels of LC3 do not necessarily change predictably upon autophagy induction. Induction can lead to an increase in the conversion of LC3I to LC3-2, but if lysosomal turnover is rapid, it can also lead to a decrease in LC3-2. Therefore, the simple detection of LC3-2 alone is insufficient to monitor autophagic flux. It must be measured with and without a lysosomal inhibitor to truly assess the rate of degradation. Finally, while LC3 is commercially available and a key tool, it is vital to remember that LC3 can be associated with non-autophagic structures, such as vesicles involved in specialized endocytosis and non-conventional secretion systems. This underscores the necessity of combining LC3 analysis with morphological and functional assays to achieve accurate and reliable conclusions in autophagy research.
We invite Mrs. Svetlana Kasimova to speak. The topic of his speech is sleep disorders in adolescent age.
Good morning, esteemed colleagues, ladies and gentlemen. My name is Svetlana Kasimova, and I am here today to present our research focused on the peculiarities of sleep disorders in adolescents and the critical factors that influence their occurrence, including chronotype, sleepiness, character accentuations, and stress. Adolescence is a pivotal life stage. It is a time when the ability for abstract thinking, analysis, and independent problem-solving fully forms. However, this period is also characterized by a strong internal drive to define one's identity and find one's place in society, a process that requires establishing relationships and mastering various social roles. Sleep is an integral and essential part of an adolescent's life, regularly occupying about 1/3 of their time. It is controlled by complex brain structures like the medulla oblongata, midbrain, pons, and cerebellum. Critically, the circadian rhythm determines the daily cycle of sleep and wakefulness, while the ultradian rhythm dictates the sequence of slow-wave and REM sleep phases. For adolescents aged 13 to 18, the total recommended duration of nocturnal sleep is a significant 8 to 10 hours per day. Unfortunately, sleep disorders in adolescents are a common occurrence and carry serious consequences for both physical and mental health. The causes are diverse, ranging from external factors like changes in schedule and parenting style to internal issues like stress, fatigue, and physical problems such as active growth and the menstrual cycle. We often observe specific conditions like delayed sleep phase syndrome, which arises from irregular circadian rhythms and causes difficulties with both falling asleep and waking up. Furthermore, parasomnias like sleepwalking and night terrors are often triggered by factors like insufficient sleep or traumatic events. Diagnosing and treating these complex disorders necessitates a highly individualized approach. Key factors and predictors of sleep issues. Several key factors strongly influence an adolescent's vulnerability to sleep disorders. The adolescent's inherent chronotype, whether they are a morning lark or a night owl, can be associated with the risk of developing sleep problems. Lifestyle factors such as the use of medications, internet addiction, and constant use of gadgets are significant disruptors of natural sleep patterns. Crucially, psychological factors such as stress, anxiety, and depression directly impair the quality and duration of sleep. Other contributing causes include repetitive thinking before bed, social pressure, stringent school demands, weather factors, noise, and generally unfavorable sleeping conditions. To thoroughly investigate these relationships, we employed a robust research methodology. This included Schmieszczek's questionnaire test to assess character accentuations and identify risk for mental disorders, the Epworth Sleepiness Scale to measure daily sleepiness, the Hornostberg Morningness-Eveningness questionnaire to determine chronotype, and a specialized questionnaire to assess the predisposition to stress development. The results of our study provided clear and important correlations between psychological traits and specific sleep disorders. Firstly, we found that insomnia symptoms in adolescents strongly correlate with symptoms of other sleep disorders. This confirms the idea that insomnia can function both as a standalone mental disorder and as a symptom secondary to other underlying sleep disturbances. Secondly, specific character accentuations demonstrated close links to sleep pathologies. The anxious, dysthymic, pedantic, and excitatory accentuations are closely correlated with disorders such as insomnia, night terrors, sleepwalking, and social jet lag. The cyclothymic accentuation type, on the other hand, correlates specifically with circadian disorders and cataplexy. Finally, the study observed that in younger adolescents, disorders such as sleepwalking and night terrors are the most frequently encountered, a finding that aligns with the established age-related distribution of these specific parasomnias. In conclusion, our findings emphasize that successful management of adolescent sleep disorders requires an approach that goes beyond addressing sleep hygiene alone, deeply integrating an understanding of the individual's psychological structure, stress levels, and natural chronotype. Thank you.
We invite Mrs. Maria F. Mangajas to speak. The topic of his speech is the lacuna and latency of Filipino maritime anthropology.
Good morning, everyone. My name is Maria F. Mangajas, and I'm addressing a crucial paradox in Philippine social science, the surprising scarcity of maritime anthropology in a nation defined by its extensive coastline and reliance on the sea. The Philippines is an archipelagic nation, comprising over 7,000 islands. Over 60% of its population of more than 100 million resides in coastal areas, making the fisheries sector a critical component of national life. This extensive marine domain faces compelling, market-driven pressures, including overfishing, resource degradation, pollution, and ineffective regulation. Despite this overwhelming demographic reality, anthropological studies focusing on this maritime aspect are surprisingly scarce. As early as 1998, Filipino anthropologist Ponciano Benigin noted the ironical poverty and youth of maritime anthropology in the Philippines relative to the well-established upland and lowland ethnographies. This yawning gap in Philippine social science is starkly evident in the publication record, where non-Filipino authors surpass local scholars in books and academic journals on coastal fishing communities. This disparity prompts a crucial question. Given the nation's reliance on aquatic resources, why has the surrounding sea not played a larger, more central role in the development of Philippine anthropology? Current conservation efforts, such as establishing marine protected area networks, highlight this deficit. We have far more information regarding fish biology and marine species behavior than the social identities, subsistence strategies, exchange networks, and cultural knowledge of the human inhabitants who depend on these environments. Intensification and decline in Philippine fisheries were already apparent in the 1930s, and today, fishers routinely report having to travel farther out to sea for greatly diminished catches. This raises the urgent research question of why fishing persists as a livelihood option despite demonstrably uneconomic returns. The paradox deepens when we realize that a substantial amount of well-written, theoretically framed, and sensitively nuanced ethnographic material on Filipino coastal communities already exists, but it remains largely unpublished. This body of work, often referred to as gray literature, comprises master's theses, PhD dissertations, and technical research reports from long-term initiatives like coastal resource management projects. These hidden resources offer rich insights into social aspects such as sharing behavior among fishers, mobility, local knowledge, and cultures of resource use and abuse. The reasons for this latent body of knowledge are multifaceted, relating to the shifting prominence of anthropology against other disciplines, compartmentalization between the social and natural sciences, and the thematic priorities within Filipino anthropology. Early formative experiences, like the anthropology field school in the 1980s, which aligned with a nationalistic drive toward social science indigenization, helped push some students toward coastal resource management and maritime anthropology. The subsequent employment of anthropology graduates in projects like the first CRM project in Lingayen Gulf further contributed to this unpublished ethnographic material. A major recent development is the work of Cynthia Neri Zayas, who established a sea-oriented anthropology program with a clear publication-oriented focus. The knowledge being surfaced through this new emphasis on maritime contexts reveals a conscious intention to indigenize ideas, privilege local knowledge, and claim one's own grounded practice in Filipino anthropology, echoing broader themes of counter-hegemonic discourse that have recurrently stimulated Filipino scholarship. We must conclude with a call to action. Until Philippine universities commit to the digitization and open online referencing of these theses and dissertations, a significant volume of valuable ethnographic material will remain underutilized and out of sight, perpetuating the very knowledge gap we seek to close. Thank you.
So, we sincerely thank all participants of the Second International Scientific Conference, InterSci 2025, Global Innovations in Modern Science. Special thanks to Petra Buric, Snezhana Babic, and Iva Blazevic from the Juraj Dobrila University of Pula for their tremendous work in organizing and hosting this successful event. We invite everyone to our next conference. Thank you. See you soon.